Good morning, everybody. Good afternoon for people that are in other places of the world. Very excited to be here today. This is going to be unfortunately, my last live broadcast for a little bit over a month because I'm about to go to Ecuador for a month where I'm going to be hanging out with Nancy Miarelli, who you may have remembered if you've been watching these live videos for a while. I'm going to be doing some videos with her about her tour service, her insect safaris that she does in Ecuador. It's going to be really cool. Hopefully, I'll be able to help her get that business really off and running. She has a lot of tours booked for the summers, but hardly anything booked for the winter. And I don't know why that is. I mean, why don't people want to get away from the horrible cold and rain of wintertime and go to Summerland? Doesn't make any sense to me. So yeah, hopefully I'll be able to help her promote her tours for winter seasons, and maybe next year you guys can book a tour and I'll be there because I'm planning on probably going down there once a year every winter to get out of the cold here. Now that I'm in Canada, it's going to be very nice to escape to Ecuador every year. So today's discussion is going to be on punctuated equilibrium. And real quick here, just seeing if there's any questions before I start here. Punctuated equilibrium is a topic that a lot of people have been very confused about. Let me share my screen here and my slides. So here we go. What is punctuated equilibrium? And punctuated equilibrium is the observation that species can be morphologically stable for many generations, and then they'll undergo relatively fast morphological change over just a few generations. And this is something that, Darwin noticed that evolution doesn't happen at a constant rate. That it can speed up and slow down, but Stephen Jay Gould is the guy that really paid really close attention to this. What's actually going on here? Why is it that evolution happens in these kind of spurts, and then morphologically evolution seems to freeze within populations? And he noticed this by studying the fossil record. I believe he was he did a bunch of work with snails. I think is actually what really helped him come to this conclusion of punctuated equilibrium. And when he wrote a paper on it, it became really popular. And there's actually a lot of debate. Not so much that punctuated equilibrium is real. It's definitely a thing that happens, but how significant it was. So, if you were around back then, you might recall him doing a lot of debates with Richard Dawkins. Richard Dawkins basically he just didn't like how punctuated equilibrium was seeming to get so much attention when it was just a really what he thought is just a minor aspect of evolutionary biology. But I actually disagree with Dawkins on this, and I think that Stephen Jay Gould really did point out something that's interesting and worth thinking about and worth studying. So again, he observed that species will be stable for many generations, sometimes millions of years, where every fossil that you find of this organism, you can tell that it's definitely this one particular species. And then you'll get relatively fast morphological change over just a few generations, and all the fossils that you find after that, you can tell are this different species. And so we're going to talk about the cause for stability. Why is it that species stabilize at certain times during their history on Earth? And why is it that they change rapidly at other times? And there's massive confusion about this. So first of all, punctuated equilibrium is a little bit confusing just to begin with, but then you've got the professional misinterpreters out there who have been writing books and saying that punctuated equilibrium is the equivalent of the evolution of hopeful monsters, or that all the change happens immediately. All the evolutionary change happens immediately. That's not what Stephen Jay Gould said happens. That's not what we see in the fossil record. We see lots of transitional species. That's another thing that people say that Stephen Jay Gould says there are no transitional species, and this is actually Stephen Jay Gould responding to that. He says, "Since we propose punctuated equilibrium to explain trends, it is infuriating to be quoted again and again by creationists, whether through design or stupidity. I do not know, as admitting that the fossil record includes no transitional forms. Transitional forms are generally lacking at the species level, but they are abundant between larger groups." That's Stephen Jay Gould. Sorry, my dog's barking. I gotta see what's going on here. Sorry about that. So Stephen Jay Gould was, he was constantly misquoted, and this idea of punctuated equilibrium was constantly used by creationists to try to misinform the public about how evolution works, and that was super frustrating for him. It's actually a little bit his fault in that he used wording that was really easy for the shysters to take advantage of. One of the weird things about teaching evolution is that not only do you have to be really careful not to teach things in a way that will confuse genuine students, but you also have to teach things in a way that, there are these people that are professionally lying about evolution for a living. And you got to be careful so that you don't say things in a way that they can easily use to misquote you. And, people now have to be extra careful because of this. So that's kind of frustrating. So they say that the creationists will say that there are no transitional forms. So this species here, can anyone guess what this is? Maybe type what you think this is in the comments section. It's a giant meat-eating dinosaur with little stubby arms that just have two little fingers on there. A lot of us, I think, would assume that this is a T-Rex. That's actually not the case. This is Albertosaurus. This guy here, same thing. Giant meat-eating dinosaur, tiny little stubby little arms with two fingers. You might think it's a T-Rex, but no, that's Gorgosaurus. This one here definitely looks like a T-Rex. Giant, massive head there. But again, that's not T-Rex. That's Tarbosaurus. This guy, by now this must be T-Rex, right? You think this has to be T-Rex. And no, that's Dasplatosaurus. There are dozens and dozens of species of tyrannosaurids. I shouldn't say dozens and dozens. There's actually, I think, about a dozen species of tyrannosaurids that we've discovered and named, and they're distinct species, and to the untrained eye they all look the same, but when you really start paying attention, you can see here on the left we have Tyrannosaurus, and on the right we have Gorgosaurus, and you can see that there's differences in the skull width, and also Gorgosaurus has these little I guess bumps over his it's actually over his not his eyes, and there it's actually in front of his eyes. And Gorgosaurus would have seen more side to side, whereas T-Rex would have looked straight at you if he's looking on at you like he is in this picture here. So there are these subtle differences between these species, and we can tell that they're different species because of these subtle differences. There actually is some debate over a couple of them. We think that they might just be variations within one really diverse population, or sometimes there's a couple of tyrannosaurids that we've named and then later discovered that they were probably just juveniles of a different species. So that happens sometimes in paleontology. When you're trying to figure out who is who just by looking at bones, it can be very tricky, especially considering the fact that a lot of these animals, we don't have entire skeletons for them. In fact, there's no 100% complete skeleton of T-Rex. We've got, I think I've got a slide about this later on, but we've got about 50 specimens of T-Rex, and only 7 of those are 45% complete or more. So the vast majority are just, a few teeth or a bit of the jawbone, chunk of the skull. And in that case, it's actually kind of hard to even diagnose what species it is. We kind of go based on, we go based on the skeletons that are really well formed. That's what we use to really define what the species is, what its traits are. And then when we find these smaller chunks of skeletons, we can use the descriptions that we already have of different species to try and figure out what those teeth belong to, and so on. So when people say that there are no transitional forms, well, they are lying. There are many transitional forms. There's like I said, there's multiple tyrannosaurids, multiple tyrannosauroids as well. On the bottom here, we have a few tyrannosauroids that are on display here. And they span across from 165 million years to 66 million years ago is kind of the time span of the evolution of tyrannosauroids and tyrannosaurids. Tyrannosauroids came first, and they evolved from more primitive theropod dinosaurs. And kind of the big separation is that the tyrannosauroids have 3 fingers typically. There are some exceptions. Some of them have 2. Some of the ones that only have 2, we think actually might be juvenile tyrannosaurids. And then we have the tyrannosaurids, which have the 2 fingers, and in general they're larger than the tyrannosauroids. And we see this really nice. I mean, it's a really nice picture of evolution. You see the more basal theropod dinosaurs coming about first. Then you see the tyrannosauroids. Then you see the tyrannosaurids, and eventually at the end you get T-Rex, the T-Rex that we all know and love from the movies, right? And during this time, though, so we do have all these transitional species, but during this time, each species stays pretty much unchanged. So T-Rex, again, I mentioned that we have 7 species, seven specimens that are over 45% complete. And when we look at them, they span about 2 million years in Earth's history. And when we compare them to each other, we see that they're not. There definitely are size variations, and there definitely are morphological variations. Some of them have more or less teeth than others. But it's really hard for us to tell when we're looking at just 7 individuals if those differences are just differences between individuals in a population, or if those differences are evolved differences. So it gets pretty tricky when we're just looking at bones. But for the most part, it seems that T-Rex, during those 2 million years when T-Rex was around, T-Rex wasn't really changing in any sort of a direction. Pretty much the animal was staying the same morphologically. And yeah, here we go. I'm just talking here about the 2 million year time range, and then there are only 7 specimens that are more than 45% complete. T-Rex is also extremely similar to Tarbosaurus. Tarbosaurus is an Asian species. T-Rex is an American species. And, Asia and America were connected, not during the time of T-Rex, but before the time of T-Rex, the 2 continents were connected, and the population split. And so some researchers actually put Tarbosaurus as a Tyrannosaurus. Some of them say that it's its own thing. There are some morphological differences, but they're so insignificant that a lot of people have argued, hey, this is the same species. It's just been separated for, maybe not very long. The 2 populations have not been separated all that long. Maybe we should consider them just one genus, actually. So just Tyrannosaurus. So there is debate in paleontology for some of these transitions, but for the most part, a lot of people think that we should have this as its own genus, Tarbosaurus as its own genus, and Tyrannosaurus as its own genus. Sorry, I think I said species earlier, but when we're talking about fossils, we talk about genera instead of species, because we can't get specific enough to determine individual species. So this is the pattern that we're seeing in the fossil record. We are seeing these nice gradual transitions, but it's like it's like the transitions happen in ticks, and then they'll stay the same for a while. And there's another tick, and they'll stay the same for a while. So what's causing the stability of species? Why is T-Rex pretty much unchanged for 2 million years, at least 2 million years? I mean, T-Rex probably existed longer than that. It's just 2 million years is the time span from the oldest specimen to the youngest specimen that we currently know of. Well, one of the causes for stasis, for equilibrium, is the fitness landscape hypothesis. So the fitness landscape hypothesis is that, you know, within any environment, there is an optimal version of the types of animals that currently live in that environment without dramatically changing them. So there's a bunch of tiny little mutations, slight changes that could happen to make them more fit within their environment. And so here we have a fitness landscape that's got 3 peaks, and you see the population starts at the bottom of this fitness landscape. So early on, the population is not very well adapted to this landscape, but over time they end up climbing this fitness landscape, and they end up settling on a peak on the right here. And so this peak represents the phenotype, the characteristics of an organism that would be best fit for their environment. And as we're seeing here, there's 3 possible peaks that they could have climbed, but once they're on a peak, it's hard to get to another peak. So this actually forces them to stay morphologically similar to each other once they are optimized to survive and reproduce within their specific environment. And this, not only is this just something that people speculate with, this is a mathematical model and, you know, got this computer model here, but this is actually something that we observe in real life experiments with evolution. These 2 graphs here show the fitness of bacteria in the long-term Linsky evolution experiments. So what we're seeing, this graph, the graph B is the most interesting here. This is zoomed out. So over 50,000 generations of bacteria, and actually by generations here, he's talking about, I believe he's talking about transfers from one medium to another, not just individual generations of bacteria, but I'd have to reread the paper to know for sure there, though. But what we see is that, so he took a bacteria, a starting bacteria, which was not adapted for his lab. He put it in a very specific environment in his lab, and then he watched it evolve over time. And it evolved rapidly at the start. And then, as we see, its evolutionary changes slowed down. And by the time he reached 50,000 generations, the evolution has almost leveled out. It actually never levels out. It keeps climbing forever, but it slows down exponentially, and the amounts of changes that can happen. And what's going on here is that early on, that bacteria is not very fit at all for its new lab environment. And so there's tons of easy mutations that can happen that will make it more fit for its environment. It's very easy for it to adapt to its environment. And it's very easy for a mutation to make one of the individuals better at surviving and reproducing than its neighbors. And when that happens, it's going to take off. It's going to consume most of the resources, and it's going to end up having more offspring than its comrades in that environment. And so early on, there are lots of opportunities for evolutionary change that's going to be beneficial. But as the bacteria gets more and more fit for that specific environment, the chances of a beneficial mutation become smaller and smaller. And the reason for that is that it's so well optimized already within its major body plan, within its major metabolic structure and so on, that most mutations end up making it less efficient than its neighbors rather than more efficient than its neighbors. Early on, most mutations made it more efficient than its neighbors. Later in this graph, the majority of mutations make it less efficient than its neighbors. And so those mutations end up getting lost. And this is a very clear example of bacteria evolving and getting stuck on the top of a fitness peak within their current environment. And I want to jump to questions real quick here, because this concept is might be new for a lot of people. So let me just go back to see if there are any questions. And if you do have questions, just go ahead and do @statedcasually so I can see that in orange. Oh, shoot. You guys are not seeing my slides that whole time. Oh, wow. That's not good. I'm sorry about that. It's a major fumble. All right, well, I think I'm going to have to edit out this video before publishing it afterwards, but let me show you this fitness landscape here. Here's the fitness landscape, and you see these 3 peaks, and you see the population evolving onto the peak on the right here. So this is in a static fitness landscape. So this is assuming that your environment is remaining constant. And what's going to happen when your environment remains constant is the bacteria are going to find just one, or the organisms will find just one morphology that works really good in that environment, and they'll get stuck on that fitness landscape. And it's really hard for them to transition to another morphology when they're currently at the top of a fitness landscape. Hopefully that makes sense. Let me go back to questions here. Let's see, show us the graph. So I'm showing you the graph right now. Hopefully you can see that. Yep. So here, this graph here, graph B, is the one that shows us this leveling out of the fitness of the bacteria here over time. So we start out at generation 0, and that's the starting population that they started with their evolutionary experiment. And you see that early on in that those experiments, pretty much every mutation that happened, not every individual mutation. Obviously, there were lots of individuals that would mutate and they would end up spiraling out of control and go extinct. But there were lots of chances for beneficial mutations because this was a new environment to that bacteria. But then once that bacteria has been stable for a while, they've been living in that environment, they've been subjected to those selection pressures for a long period of time, well, then they just stable out right there. Let's see. Someone, Vienna asks, are coelacanths an example of slowed down evolution? Coelacanths are sort of. So coelacanths, modern coelacanths are actually dramatically different from fossilized coelacanths, but they are still recognizable as coelacanths. So what you've got going on there is you have a body form that is really well adapted for their particular lifestyle, and you have populations splitting off of the coelacanths and evolving into other types of organisms, but that main population stays there. And I'm actually going to talk about that in a minute here when I talk about how it is that new species form, how it is that new species evolve. Let's see. Any other questions before I go on? All right, continuing on. So you are now seeing my slides here. So, by the way, the graph here on the left is zoomed in, so we see that fitness actually levels out, and then there'll be a beneficial mutation, and fitness jumps, and it jumps quite dramatically. I should also point out that another way that you can get a dramatic jump, and I should have included this graph too, which is, I think it's also in that same paper. By the way, there's links to this paper in the video description. But another way that you can get a dramatic jump is by a mutation that ends up being extremely beneficial. So in one of the long-term evolution experiments, a mutation caused the bacteria to be able to eat a new type of food. And there was just a rapid explosion in fitness. And it caused the graph to do this again. So you have this big jump, this time where most mutations end up being beneficial, and then it starts to level out, and then it jumps again, and then it levels out. And that second jump in the case of the Lenski experiment was an adaptation to a new food that was in the environment. But those types of jumps are rare. The types of jumps from some sort of internal mutation are pretty rare. Normally, organisms will just adapt to their environment. They'll end up on the top of a fitness peak within their fitness landscape that they happen to be evolving inside of. So you can also have what are called dynamic fitness landscapes. And this is just where the environment is changing rapidly. Maybe you have the introduction of a new predator, and then that predator ends up going extinct. And then, so you're evolving back and forth between, different parameters. Maybe you have a weird drought cycle. And this is another thing that will also keep a species pretty much looking the same throughout evolutionary time, because they actually spend more time between two peaks than at the top of either peak. And because the fossil record is so horrible, I mean, we've got T-Rex was around for 2 million years. We only have 7 specimens that are really any good. So because our ability to actually study these organisms in general is so poor, that's not always the case, but for most organisms that is the case. It can be really tricky to tell what's going on. Are they stuck on one fitness landscape, or are they bouncing between two changing fitness landscapes? It's difficult to tell. This here is a species of finch from the Galapagos, and these are actually 2 individuals from the same species. And now, one of the interesting things about punctuated equilibrium is that genetically organisms are evolving constantly. So mutations happen at a constant rate, but morphologically, mutations or changes seem to happen in these spurts. You have these freezings, and then you have these spurts. And so what's going on here? Well, within any population, there's a lot of genetic diversity. These 2 individuals are from the same population, and they have dramatically different shaped beaks, right? And when a population is successful, when it's growing, when it's expanding, it will increase in genetic diversity, and it will increase in morphological diversity. But the average finch here, I wish I had a picture of the average. The average is actually kind of in between these 2. And so if you're getting a random sample of finches, you would almost never find one with a beak this small or one with a beak this big. You would almost always find one with its beak right there in the middle. And this is one of the problems that we have with the fossil record, is that the fossil record is we're getting a tiny, tiny fraction of the actual population. And because of that, we usually get the morphologically normal individuals. The morphologically abnormal are less common, of course. And so we can't see the incredible diversity that actually exists in a population. It's very rare that we actually see that. Well, in the case of these finches, there was a selection event in 2003. There was a drought, and it ended up killing all of the large beaked organisms. And what that meant is that these small beaked individuals became super popular, or became super common in this population. And if we were looking at this in the fossil record, it would have looked like a fairly dramatic change. There used to be medium-sized beaked finches, and now there's only small beaked finches. And that's because all the large ones, and actually a lot of the medium-sized beaked finches, they immediately starved that year because of this drought, which changed the types of seeds that were available. And they were also under competition with another large beaked finch that was on that island. and it was more efficient at getting the few large seeds that did exist. And so we see this rapid evolutionary change. And this helps us understand how it is that rapid change happens throughout evolutionary history. So one way that rapid change can happen is you have a diverse population and it gets whittled down by natural selection so that all the weird traits that happen to be in that population are now the norm. And so you see this oftentimes as a sudden change in the fossil record. The other way that this can happen is through peripatric speciation. So you start out with an original population of, let's say, tyrannosaurids, let's say Gorgosaurus, for example. And then a small group gets split off from the main group, and they no longer can breed with the main group. There's no more gene flow. So when you have a big, large group of sexually reproducing organisms, they're constantly sharing genes with each other. So if part of the environment is cold and it would be beneficial to evolve traits for a cold climate, that actually can't happen when the population is well connected, because all of the warm genes that are being selected for in the rest of the environment keep flowing into the colder environment. So you get no morphological change. Even though there is selection pressure in that colder environment for new traits, you don't get them changing in any sort of a directional way. They just remain stuck in the normal morphology of the rest of the population. But once you get a population that splits, then all of the mutations that happen in that new population and all of the best traits that happen to exist in that population already, all the best genes for that colder climate, well, they can all get selected for. And there's not going to be this interference from the rest of the population where the warm adapted genes keep on leaking in to the new population. And this is one way that new species form. And because this often happens with a small population that breaks off from a larger population, what we see in the fossil record when this type of speciation happens is we see one species or one genus rather, and we see that it's constant across sometimes millions of years. And then we see all of a sudden the appearance of another population that is slightly different, but different enough that it needs its own genus name. And sometimes we can see that new genus live parallel to the older genus for a period of time, and sometimes it'll actually take over. Maybe it's got some really nice adaptations that allow it to eventually take over. But we'll see, a lot of times we'll see two species that are clearly related, two genera that are clearly related, but one appears later, and then they live parallel to each other for quite some time. That actually happens a lot with tyrannosaurids, for example. If I go back to my T-Rex species, tyrannosaurid species. By the way, I think you guys missed this. This on the left is T-Rex. On the right, we have Gorgosaurus. These two species did not live parallel to each other, but this guy, the Spletosaurus, did live alongside Gorgosaurus. And Gorgosaurus was a lot smaller and probably could get around faster. Whereas the other guy, this big, bulky animal, might have been preying on different items or maybe doing more scavenging. Very bulky, very heavy animal that probably could not move as fast, nearly as fast as Gorgosaurus. And they come up parallel to each other in the fossil record. So this appears to be an example of peripatric speciation. So one of the things that critics of evolution have been writing is they say that this is all just an excuse because there's not good transitions in the fossil record. And so Stephen Jay Gould made up this whole concept and all these concepts of fitness landscapes and peripatric speciation. These are all just made up to explain away things in the fossil record. Well, that's not the case. We actually have very clear fossil records in some cases. So this is a group of diatoms, and diatoms are really neat because we can actually watch their evolution happen almost perfectly. because diatoms, they live in the water. They're these small little microfossils. They have these glass-like skeletons that they form around their cells. They're single-celled organisms. But they form these shells that fossilize really, really well. And when they die, they just kind of rain down on the bottom of the lake or the ocean where they were living. And so they form this perfect fossil record, nearly perfect fossil record. And we can actually look and study these fossils, and we can see every single transition that occurred. So this transition that we're looking at here is from a species called Niagarae, evolving into the Yellowstonensis species. And we see this evolution happening step by step in the fossil record. And what happened here, the Niagarae species is very common. It's all across North America, including around Yellowstone. And what we see is we see one little population of this species, which, by the way, it's a species that's still alive today. We see one little break off of this evolve into the Yellowstonensis species. And it's hard to tell if you're not trained looking at these what the differences are between these two species, but they're actually quite clear once you start learning how their morphology works. You count the little lines and you count the spikes, and there's a bunch of really obvious differences once you start studying it. But we see this transition happening very smoothly in the fossil record. And this is peripatric speciation. This is a tiny group, got broken off from the main group, and it happened to go into an area that was a warmer climate. And because of that, we can actually, by looking at pollen in the area, we can tell that the climate got warm fairly rapidly and changed quite a bit. And that difference in selection pressure drove the difference in evolution of this species. So, punctuated equilibrium was this idea that Stephen Jay Gould came to by looking at the fossil record. And we came to by looking at the way in which new species form, different models that different researchers had put forth about how new species form. And then we had it confirmed by looking at fossils like this, where we can actually see all the different individuals and all the nice little smooth little steps. And so we see that evolution actually does happen exactly like Stephen Jay Gould had predicted. And this is why I say that I think it's really important that we understand punctuated equilibrium and take it seriously, as opposed to Dawkins' original argument that it wasn't really all that significant. This is a prediction that he made looking at fossils and has now been confirmed when we look at microfossils such as diatoms here. I want to talk about how it is that something like this could happen today. These are elephants from the Oregon Zoo, and I used to volunteer there. And this one kind of in the middle here, looking down at the pumpkin, this is Shendra. And she is an Asian elephant, just like all the other ones. But if you notice, she is tiny compared to them. And she's also very, very hairy. And she's actually a full adult. She's full grown here. But she's just a different population. She's what we call a pygmy Asian elephant. She's from a population in Borneo that's just smaller and hairier than most other Asian elephants. She's not a unique species. She can breed with other Asian elephants and have offspring, but she's very unique. And if we were to imagine a scenario where the Earth suddenly got cold, and you've got a population of Asian elephants that are very diverse. There's extreme diversity in ear size. There's extreme diversity in the amount of hair. Here I've got a extremely hairy elephant. They're actually hairier when they're babies, and there's a gene that causes them to lose their hair as they get older. But the pygmy elephants don't lose their hair nearly as much. So there's this dramatic variation in elephants already. If you have smaller ears, you're more adapted to the cold. If you have more hair, you're more adapted to the cold. If you have more body fat, you're more adapted to the cold. And right now, all of these traits are spread out among elephants. But if we had a rapid change in climate, that would act as a funnel. And all of the elephants that had the slight ability to survive better in the cold would end up surviving and reproducing better than the elephants that did not have those abilities. And what we would see is we would see that eventually the offspring of the extra hairy elephants. So let's say that there's some elephants that have the gene that causes the hair loss to occur. Maybe that gene breaks in one elephant, and so you get these hairy elephants up into adulthood. Those end up being more successful, and they end up breeding with the elephants with small ears, which are also better at surviving in the cold. And those end up breeding with the elephants that have extra body fat. And so you start out with a population with a bunch of individuals that are slightly adapted already, just happen to be slightly adapted to a colder climate. And their offspring end up meeting each other and creating these basically like superhero elephants that are super well adapted to their environment. So natural selection acts as a sieve, taking all the variation that currently exists in a population. And when the climate changes, when the environment changes, maybe there's a new predator, maybe there's, in this case, the climate changes, all of that gets bred into a few individuals, and they end up just absolutely thriving in this new environment. And by the way, this is a website where you can go and learn all about the morphology of diatoms here, and it's a pretty cool little website. I got links to that in the video description. The last thing that I want to say here is that if you care about the math, you want to know the numbers about how this stuff works, there's this great paper called The Probability of Fixation in Populations of Changing Size by Sarah Otto. And basically what she finds in her models is that you get rapid evolutionary change in two circumstances. When the selection forces in an environment change, you get rapid evolutionary change. And that's because natural selection is, weeding out, it's whittling down the population. You also get rapid change when a population is in a state of exponential growth. And this is because when a population is in a state of exponential growth, lots of mutations are happening because the population is expanding. And mutations don't just happen from parent to child, they also happen sideways, right? So the bigger the population is, the more mutations are occurring. And because if you have a mutation that makes you 1% better at surviving and reproducing, 1% doesn't sound like that big of a deal. But when a population is in a state of exponential growth, that 1% is an exponential advantage. And so you actually get that 1% taking over the population a lot quicker than you might expect. And she goes over this math in her paper here and explains how this works. It's really fascinating stuff. If you care about the numbers, check out this paper, The Probability of Fixation in Populations of Changing Size. And that there's a link to that in the video description here. So that wraps it up. I apologize for the lack of visuals during the first part there. There I am again. And if there are any questions, I would love to see what those are and address those now. Jay asks, have you seen the reimagined images of the really fat T-Rex with the justification that they should look more like chickens, since we don't know exactly what the muscles and fat was like? I've seen some pretty stocky T-Rex images. I don't know if I've seen the one that you're talking about there. But yeah, one of the big problems with paleo art, of course, is that we have no clue for a lot of these organisms what they look like aside from their skeletons. And there's a really good book, I think it's called All Your Yesterdays by John Conway. He's a paleo artist, and he redoes modern animals in the way that artists typically redo dinosaurs. And it's really funny because he's got like a jaguar, and you can see all the bones in its skull, and it looks nothing like a jaguar, right? So there is that problem in paleo art. where we don't know what the heck the soft tissue looked like on a lot of these organisms. There are some exceptions to that. There are some fossils that have really nice outlines of what their bodies actually look like, but it's pretty rare. Lone Wolf says, talking about elephants, what's your thoughts on the attempt at de-extinction of mammoths? I actually have a video about that, where I talk about, can we bring dinosaurs back? And I talk about the mammoths. I think that the mammoth thing, I think that in their attempts to do that, there's probably high probabilities that they're going to screw up a lot, and that's going to be unpleasant for the elephants involved in those experiments. But the Asian elephant is actually really similar genetically to mammoths. There's the mutations that have occurred, the differences genetically are surprisingly small. And so they think that we can kind of, maybe using CRISPR, or just maybe even just selectively breeding elephants that already exist, we can get something that looks like a mammoth again. It wouldn't be an actual mammoth exactly, but it would have the traits. It'd be hairy, and it'd be bigger. It'd have smaller ears, and so on. I think that would be cool. I would pay money to go see it once it's done, but I don't think that I would not. It's not something I would do. I think it's got a lot of potential for hurting elephants in the present, and there's probably better things we could do to spend our money when it comes to restoring habitats and helping endangered species that are alive, helping them survive. But it would be pretty epic to see a giant woolly mammoth. Let's see. Any more questions about punctuated equilibrium? Well, I think that does it. Again, this is going to be my last video for about a month, I would say. I'm going to be in Ecuador, but while I'm there, I'll be filming. So, provided that I come back, I'll have lots of videos to share with you. So I'm excited. It's going to be fun to hang out with Nancy. It's going to be fun to go get in the sunshine and the jungle and, learn about insects for a while. So long for now. I apologize for screwing up the slides early on. Let me see. There's one more question. Could humans as a species be moving towards a punctuated equilibrium if medical technology keeps going, keeps improving? So right now, humans are actually, until recently, human populations were actually evolving quite rapidly. So humans are in a state of exponential growth. And because of that, any beneficial mutations that do occur end up kind of dominating the population quicker than you might expect. And that's what's allowed, different races to emerge that are very adapted to the different environments where they lived, because humans are in this state of exponential growth. But now we are all mixing back together, and we are, we have medicine that's dramatically changing how it is that we are evolving. But what's going to happen is medicine really isn't hurting us. There are some people that say that medicine is hurting our evolutionary trajectory. I do not agree with that. Medicine is allowing us to increase in genetic diversity, and that's a good thing, because you want your population to be nice and genetically diverse, so that when there is another selection event, and there could definitely be another selection event, severe selection event in the future, you have a lot of diversity, a lot of different traits in that gene pool that can then be whittled down by natural selection and form something new. So we are in this state of exponential growth. We are still evolving. If you look at human populations in the past, they were different than they are today. But medicine is kind of slowing things down. Medicine is slowing our ability to evolve in any specific direction. We're basically just becoming more diverse right now, and it'll be probably another selection event that will pull us back into, or push us one way or another. Maybe nuclear war, maybe climate change might really screw things up for us. It might rapidly screw things up for us. And you might actually see humans splinter into different groups that then start evolving down different trajectories again, like they did in the past. We had this out of Africa split where you have Northern Europeans evolving in a different trajectory than Africans, and a different trajectory than Australians, and so on. What role do epidemics play in punctuated equilibrium? Well, an epidemic would just be a really harsh selection event. And there are, you can actually see there is a genetic marker for the Black Plague, for example, among Europeans. So you actually see epidemics changing our genomes and changing our phenotypes slightly as well, too. So that is pretty cool. Yeah, there are now people editing DNA, human DNA with CRISPR. So who knows what will happen there. If that ends up going anywhere like the genetic modification of food has gone, what's going to happen is that scientists will discover a couple of genes that are really cool, and everybody wants those genes, and we'll end up becoming more the same. Maybe that's not what will happen. Maybe it'll end super poorly. Maybe we'll screw something up, and only the people that have not been involved in those experiments will end up surviving. Who knows? Who knows what will happen? It's a big question mark on the future. Jay says, edit away my diabetes. If you have type 1 diabetes, that is something that could be edited away. Not in you, because we can't, well, maybe this is changing, but right now, so gene therapy is this idea that you can actually change an organism's genome when it's already fully developed. And it's difficult to do that because you have to change the genes in every single cell of that person, or every single cell that's expressing the bad version of the gene. I think in the case of type 1 diabetes, it's only a few tissue types that you actually would need to change. But trying to get CRISPR or whatever other method you're using into every single one of your cells that's expressing the problematic phenotype for diabetes, that's a difficult thing to do. So it's a delivery problem. We have trouble delivering gene therapy to living organisms. There are people that are working on this. I think they're using viral vectors right now. So they'll infect your cells with a virus that has the gene modifying molecules. But you can also, of course, treat diabetes by just making insulin artificially and then injecting that into your body or taking that in. Let's see. Fenno says, will it go the same way as GMO food with everything needing marketing and people refusing to mate with the genetically modified human? Probably. I wouldn't be surprised. I think we probably will mirror the GMO food thing pretty closely. The good things and the bad things. But yeah, designer babies probably are on the way. And here's one of the interesting things about this. There's a lot of reasons not to genetically modify humans. There's a lot of reasons not to do it. Mainly, I would say that the most important reason is that this is a new technology and we're messing with our actual genome. And it's just kind of dangerous. It's dangerous because it's a new thing and it's extremely powerful, right? But even if you have really good arguments against genetically modifying humans and you convince governments to outlaw it and so on, there's always going to be someone that does it anyways. This is kind of the curse of the progress of science. Science has done all these wonderful things for us and continues to do all these wonderful things for us, but also it's unstoppable. we see this with the invention of nuclear weapons. Like once we invented the bomb, we could not uninvent it. And actually, once we realized the bomb was possible, we couldn't stop it from being invented. In World War II, we had people telling the U.S. military, "Hey, we physicists, we know it's possible, theoretically possible, to make the bomb, and so we better do it really quick. We need to do it fast. We need to do it before everybody else does. Otherwise, they're going to make one first." And so there was this race, and this is kind of the darker side of science, is that our wisdom stays pretty much the same as humans, but our technology and our abilities to do things dramatically increases, and with great power comes great responsibility. And we don't really seem up to the challenge of being responsible with these new powers that we come up with. How can you guarantee that you do not mess up and create a lot of new problems genetically that end up to reduce living quality for your test cases years down the line? Yeah, that is a problem. We do try to predict whenever we come up with a new like not birth control method, a new like fertility method, for example. People do seriously consider what the negative outcomes of these new methods might be. So, for example, we can now use stem cells. We can turn stem cells into egg cells for women that are no longer fertile. We can actually take stem cells, I think it's from skin, and turn those into egg cells that can be viable, and you can actually, use them to make little humans. But the problem with that is that the mitochondrial mutation rate in skin cells is a lot higher than it is in egg cells. And so researchers have realized this is probably a bad idea to use this as a method for bringing back fertility in women that have lost fertility, because there's a high possibility that those babies will have health problems due to mitochondrial mutations. So the scientists that are involved in this stuff aren't dumb. We know that there are risks to doing all these things. The only problem, again, is that how do you get everybody on board? Even if there are risks, how do you get everybody to be careful? Because we can do this, and a lot of people want to do it, and so it's going to happen. But anyway, we'll see how that goes. I'm very curious as time goes on to see how this all plays out. Anyway, I'm going to close things there. It's been an hour. Again, I apologize for the lack of slides in the first part of that talk, but it's been a pleasure. So long for now. Stay curious.